In Chapter 10, we begin our discussion of gene expression, and the chapter is called From DNA to Proteins, Transcription, and RNA Processing. All types of RNA are transcribed from genes. So we have a long list of course, messenger RNA, transfer RNA, ribosomal RNA, small nuclear RNA, and so on. And these are all copied from genes. Only one type of RNA that we know of has codons, which are the instructions for making proteins. So the rest of these types of RNAs are made in the cell, and they have their own jobs in the cell. This is the structure of RNA. It's a single-stranded polynucleotide chain made of ribonucleotides. So you have, of course, the ribose is the pentose sugar, phosphate, and then the nitrogenous base. Of course, the RNA has a 2' hydroxyl group, which makes it much more reactive than DNA. It's also single-stranded, and that makes it a little less stable as well. So the structures of DNA and RNA compared. You can probably review most of this on your own, but I will emphasize the presence of the 2' hydroxyl group that is present in RNA but not DNA is what decreases stability and shortens the half-life of the RNA. Of course, you have a U, a uracil, nitrogenous base instead of thymine. And you actually have some secondary structure in RNA. In DNA, the secondary structure is the double helix, but in RNA, you actually also have a secondary type of structure. The RNA is not very stable compared to DNA. So this is a picture of the secondary structures that you tend to have with an RNA molecule. If you start off with the primary chain of ribonucleotides, due to internal inverted sequences, complementary and inverted sequences, you can have the formation of what are sometimes called hairpins or stem and loop structures. And those structures, when they form, are considered the secondary structures of RNA. You're already familiar with a little bit of this in terms of tRNA has 3 stem and loop structures to form what's sometimes called the cruciform shape. So this is your major information pathway in the cell. DNA, the genes in the DNA are copied into RNA. If it's an mRNA type, then that is used by the ribosome to make protein. But all other kinds of RNAs are formed from transcription, and they can do their own jobs in the cell. There is RNA replication in some cases, like say you have an RNA virus that can copy its chromosome. That would be called RNA replication, but that's limited primarily to virus. So this is a list of all the types of RNA that I mentioned before, and with their longer names. We are going to talk a little bit about microRNA and small interfering RNA in Chapter 12, and long non-coding RNA is a new type that was just discovered. So there's lots of RNAs, and not all of them are just mRNA. So I'm going to put a little overview video about transcription into the lecture, into the soft chalk on a different page, so you can review bacterial transcription. Basically, what you have is along a strand of DNA, you have genes, and the genes have they have a definite start and end. And when you have transcription of the gene, that means you have RNA polymerase moving along the gene, copying it into a strand of RNA. In this picture, you have probably hundreds of RNA polymerase enzymes traveling along each gene, and each RNA polymerase has its own RNA transcript hanging away from where it's polymerizing. And so when you have hundreds of RNA polymerases transcribing the same gene, you get what's sometimes called the Christmas tree structure, because it looks a little bit like a Christmas tree under the microscope, but it marks active transcription of one gene. So I would say a gene starts here and ends over here. Another gene starts over here and ends somewhere off the picture, and so on. So each Christmas tree, you have the DNA sort of crisscrossing your field of view, but each Christmas tree structure represents where a gene is being actively transcribed. You know that the direction of new nucleic acid synthesis is always said to be 5 to 3. And when we say that, what we mean is that the 3' end is where the new nucleotides are always going to be added. So the 3' end of the new strand is extended, the 5' end is fixed and are not changing, and so we say the direction of new synthesis is from 5 to 3. You can have more than one gene on DNA, of course, and the genes can be transcribed from either strand. So for a certain gene, the gene might be transcribed from only the bottom strand, like shown here in gene A, but there could be a gene nearby which transcribes from the top strand. The only rule is that the strand that's transcribed has to have the correct polarity in terms of maintaining a 5 to 3 synthesis of the new RNA. But you can have genes back to back like this that are transcribing from one strand or the other. This is the general layout of a gene. For each gene, you're going to have a promoter region. That's the region where the RNA polymerase will first associate with the DNA. Then you have the coding region. That's the part that will be copied into a complementary RNA. And then you have the terminator, or what they call transcription termination site here at the end. So that's the basic layout for any gene. There's some additional things we'll add on for eukaryotes, but this is generally the way it is. The strand that's going to be used by the polymerase is called the template strand. That will always run 3' to 5' here. The non-template strand is not used, and it's this, in this picture it's the top strand. The RNA will be copied then as a complementary copy of the template strand. So that means that the RNA transcript will be complementary to the template, and it will then have the same sequence virtually as the non-template strand, with the exception of using ribonucleotides and having uracil instead of thymine. The promoter sequences have what are called consensus sequences, and consensus sequences are figured out by comparing DNA from many genes and looking at the same regions of several genes and aligning them in this way. You can pick up on certain what we call conserved nucleotide sequences that have to be there no matter what kind of gene it is. So in this example, you have a T is common at this position, and A is common at this position, and then here it's some kind of pyrimidine. There's an A here, there's a purine here. This can be any nucleotide. This tends to be an A, and this tends to be a C or a G. So that becomes then your consensus pattern for that sequence that might be in front of a certain gene.